Good day. In this presentation, we will provide information on the bovine diarrhea virus. Our team consists of 6 members: Mohammed Gadwa, Jasmine Chetty, Laura Duplessis, Henri McLean, Laura Smith, and Erin Ivans. The best technique to be used to confirm the morphology of this virus is cryo-electron microscopy. This form of electron microscopy involves the freezing of the viral particles, allowing them to be observed in their natural state without being dehydrated as a result of negative staining. The morphology of this virus is relatively simple. It is spherical in shape, has an electron-dense capsid, phospholipid bilayer, and no visible spikes. It has a diameter of approximately 50 nanometers, but can be observed larger than this during morphogenesis.
The specific eclipse phase of replication of the bovine diarrhea virus is the time elapsed from when the virus successfully entered the cell, but no progeny virions are created yet. This replication phase will continue until new virions are made and lasts approximately 8 to 10 hours.
The bovine diarrhea virus in cattle is mainly transmitted by close contact with persistently infected or acutely infected cattle via the oral or nasal routes. Acutely infected animals only shed the virus for a short period of time, about 2 weeks, whereas persistently infected animals shed constantly in all bodily secretions throughout their lives. Persistently infected cattle will shed BVDV through every bodily fluid, including urine, feces, nasal excretions, tear fluid, saliva, and thereby not only contaminating the environment, but also potentially infecting every cow it gets commingled with.
Mechanisms to evade the host's immune system. The bovine viral diarrhea virus, or BVDV, in cattle is an intracellular pathogen, meaning they reproduce inside the cells of a host and lead to diseases. The BVDV causes a transient infection that results in an antiviral immune reaction or causes a persistent infection that is immunotolerance specific. BVDV evades the cattle's immune system by avoiding the immune response by inducing humoral and cellular tolerance via the manipulation of the host's immune response. This takes place by invading the fetus during early stages of gestation, leading to adaptive immunity. The non-cytopathogenic biotype of BVDV leads to persistent infection, which results in the inability to induce the type 1 interferon, which overall causes resistance to induction of cell death or apoptosis. Therefore, the interferon's defense was altered, improving the maintenance of persistent infection. Treatment or production of interferons results in immune suppression and higher chance of transmission, forming a natural reservoir for the virus in the host. Mutations and its types. BVDV has 2 genotypes, BVDV type 1 and BVDV type 2. The type 1 is the more prevalent one and type 2 more virulent, thus causing severe diseases. Then they are further classified into cytopathogenic and non-cytopathogenic. Non-cytopathogenic biotype leads to persistent infection. Thus the animal is presumably healthy but infected for life. Cytopathogenic biotype causes structural changes in the host cell through viral invasion leading to cell lysis and is fatal to the persistent infected host. Mucosal disease is a fatal form of BVDV induced in persistent infected cattle superinfected with cytopathogenic BVDV. The origin is caused by mutations like point mutations, gene translocation and duplication, as well as recombination of the persistent non-cytopathogenic BVDV. In other words, cattle have persistent infections due to the non-cytopathogenic strain of BVDV, but then a mutation of this strain forms a homologous cytopathogenic BVDV postnatal infection, resulting in fatal diseases.
How easily does BVDV grow in cell cultures? BVDV is a virus, and unlike bacteria, can only be grown within living cells. Different viruses grow in different types of cells. A comparative study was carried out to determine the susceptibility of 5 different cell types to bovine viral diarrhea. The titers obtained on day 8 post-infection are shown here. The results indicate that bovine turbinate and mink lung cells are the best for the culture of BVDV from these cell types. In a separate study where the growth medium was taken strictly from bovine origin, primary bovine embryo kidney, primary calf testicle, bovine embryonic trachea, and from buffalo lung, the virus titers did not differ significantly. From this, one can conclude that BVDV grows more easily in some cells and does not grow at all in some cell cultures where certain cells are used. Example, porcine kidney and equine dermal cells. The most likely virus to cause diarrhea in feedlot cattle is the bovine viral diarrhea virus.
To determine if this is not a bacteria-causing disease, a bacteria culture test may be performed. After the required growth period, the sample will be examined. A lack of multiplication of bacteria will indicate a negative result, proving that this is not a bacteria-causing disease. Laboratory tests to confirm a viral infection include PCR testing, a sensitive tool that amplifies a small sample of DNA using cycles of temperature changes to make it large enough to study. Virus isolation, whereby the virus is isolated in a cell culture using centrifugation or filtration. Virus neuralization involves the examination of a cell culture using a strong virus neutralizing antibody from recovered cattle as a reference test. And antigen detection. BVD virus antigen can be detected directly in tissue samples using immunofluorescence on acetone-fixed frozen sections.
Persistently infected cattle can be tested by sending samples to laboratories. The laboratory requests only that a fresh or a frozen ear notch and a whole blood collected in a purple top tube or a tube with animal identification on the tube or a labeled envelope is sent to the laboratory.
To preserve these samples for transportation, you need to keep them moist and cold, not frozen. If the collection of an ear notch is done, it should be put in an ear notch bag and kept at refrigeration temperature. When specimens such as swabs are transported, the transport media consists of a buffered solution and added proteins to protect the virus against activation. The samples need to be sent with a detailed description of the nature and distributions of the lesions if they are to be submitted for a histopathological evaluation. It is also very important to submit a detailed clinical history of that specific animal or herd to the diagnostic laboratory. These samples need to be transported to the testing laboratory as quickly as possible and within 72 hours after collection. When you transport these samples, you must also make sure that the sample is protected from any breaking as well as any leakages.
This concludes our presentation on bovine diarrhea virus. Thank you for watching.